Greetings, my name is Jorge Guerrero. I am a PhD student from the North Carolina A&T State University, and my advisor is the Professor Reza Sadegan. I am going to talk about the software tool we developed called MENDEL to automatically design functional DNA nanostructures. The nanostructures are built using the DNA origami technique that consists in the folding of the DNA strands by synthetically create DNA staples that maintain the scaffold in place. In this animation, we can see one long single strand of DNA, that is the scaffold. The short strands have complementary locations in the scaffold, and these are the staples. Letting these strands interact for some time, entropy does the job to allow the staples to locate a place, and the whole system obtains a shape, which in this case is one square. Researchers have been able to generate specific shapes using this technique, showing the proof of concept for a smiley face, and this is the AFM image obtained from the real object. This is another example for a star shape. This work of 2012 shows one box built with DNA origami, which can incorporate some cargo that is released when the box is opened, that occurs when some DNA segments interact with the keys of that box, opening it. With this technique, drug delivery that is released on specific targets is feasible. Here comes the question: how to design DNA nanostructures? Traditionally, DNA origami structures are designed using the tool CAD-Nano that we can see in this video. The process involves the manual drawing of the scaffold and selection of the staples. For small designs, this process is straightforward to reach simple shapes like this square. However, if we want big and complex structures, the requirement of time and effort grows exponentially. Our software aims to help during the designing process to create this kind of structures. Then, we introduce MENDEL. MENDEL receives a set of commands and generates a preview of the desired structure. The current picture shows the software running under Blender, an open source 3D software. MENDEL is a software library for automate the process of designing DNA nanostructures. It parametrically defines the shape and size of DNA. It is highly flexible because it's written in Python. The DNA scaffold grows linearly and can be folded to grow in 2D and 3D, and the output files are compatible with CAD-Nano. One 3D structure with 3 helices of thickness can be adjusted to 5 helices or 7 helices, and obtain for each case the CAD-Nano output file. So, how does MENDEL work? By using the provided commands, the DNA scaffold is grouped by adding 5, 10, or 30 nucleotides. If we are going to 2D, MENDEL can create rectangles of 5 base pair by 5 helices and can grow to 10 by 10 and 30 by 30. And comparing against the time we require in CAD-Nano, the difference is noticeable. For complex and parameterized structures, we define the parameters as variables in Python, and then using them inside the MENDEL commands. We can see this example has 1600 base pairs, and if we double the resolution, the model is 5400 base pairs, and the shape is basically the same, but bigger. Comparing against the time required on CAD-Nano to perform the previous designs shows the time saving when using MENDEL is enormous. In this example, I will show how to build the M character. Here, we parameterize our design and then build up each line as follows. First, we set parameters T for thickness, H for height, and S for diagonal steps. Then we add the left stick with the command rectUp. Then, to add the diagonals, we can use Python loops to be repeated S times, and add small squares using rectDown. For the ascending diagonal, we use the same code, but using rectUp. Finally, we add the right stick, and we finalize our model. To go to 3D, we add the strands to return the scaffold to the starting location, and then grow into the Z coordinates, adding a new layer, and then put the previous commands in a for loop and grow as many layers as we want. This is the Cardano representation of the design. We can modify some parameters and obtain different versions of the same design. MENDEL is available and is open source. You can obtain it from github.com/sbmi-lab/MENDEL. MENDEL requires Python 3. If run under Blender, use the version 2.8 as minimum. Thanks for your attention.